Alright, so let's take a look at another couple of the structures that exist inside of bacteria. One of them is the nucleoid region. So remember that bacteria don't have a nucleus, they just have a general area where their DNA is located. Remember that their DNA is circular and that it's much smaller than the amount that's in eukaryotes. Now keep in mind that there are some bacteria that have quite a few number of genes, but they have fewer regulatory sequences. There's less of what we called in the molecular genetics chapter junk DNA. Although keep in mind that it isn't actually junk. You have the telomeres, you have the centromeres. Bacteria don't have that. They just got their circular DNA, and the majority of the DNA is made up of coding regions so that it can make various proteins. Remember that outside of their circular genome, they also have a plasmid, or can have multiple plasmids that are small circular sections of DNA, and that usually these are important for us because they can contain antibiotic resistance genes. But there are other plasmids that have other genes located on them to do other various specific tasks. And remember that they can be transferred from one bacteria to another by conjugation, which is when you have the formation of a pili like this from one bacteria to the other, and then the plasmid is transferred from one bacteria to the other. Remember that there's also not just resistance plasmids, but that there's the fertility plasmids that are able to carry out, that allow a bacteria to carry out conjugation. Remember that bacteria do not go through mitosis. They go through something called binary fission, which is, remember, when their circular chromosome is copied and each of the subsequent cells receives one of those circular chromosomes. At the same time, plasmids can also be copied and given to the two new daughter cells. So how do bacteria then, how is their diversity? Well, the first thing is, remember that mutations can still happen inside of their circular genome, and that if a mutation were to occur, then the daughter cells would have different proteins being made. So it's not like there's no diversity in the prokaryotes. There is, but keep in mind that mutations are relatively rare. Although they are more common in bacteria because they have fewer of the mechanisms to make sure that that doesn't happen like exist in eukaryotic cells. What do I want to say? So in addition to mutations, though, there are a few other ways that variation or diversity can exist within a species of bacteria. The first is that you have transformation occurring, which is unique to, I keep saying bacteria, I should be saying prokaryotes, which is unique to prokaryotes where they're able to take up DNA from their surrounding area and incorporate that into their genome or as a plasmid. So that's sort of a unique ability. You can imagine a human going around and touching surfaces and cells absorbing that DNA. It's kind of a weird idea, but bacteria do it, and that's something special to them. So again, I keep saying bacteria, I'm sorry, prokaryotes. They can also go through conjugation, which is a process where you have the plasmids transferring from one to another through the pili. And then you've also got transduction, which is when a virus inserts its DNA and that gets incorporated into the genome. Here we'd be typically talking about lysogenic viruses. Remember that lytic viruses would incorporate their DNA, but very quickly enter the lytic phase and then produce more viruses that eventually burst the cell. So with lysogenic viruses, though, because their DNA is being integrated into the DNA and it doesn't immediately go into the lytic stage, then what can happen is that if the bacteria divides and goes through binary fission before it enters the lytic phase, and now both cells have a copy of that gene, and if it remains in a lysogenic phase for an extended period of time, these genes can stay there and eventually be incorporated into the genome. Endospores are a special structure that bacteria can form, or prokaryotes can form. A good example of this is the anthrax bacteria, and what anthrax, you guys have probably heard of this before, that they use it often in terrorist scares and stuff like that, the whole white powder thing. What anthrax can do is form this special structure called an endospore, which makes it resistant to various environmental factors. It's sort of like a protection phase of its life cycle. You know, there have been some bacteria which we've been able to say, you know, have been in this endospore phase for a very long period of time. Some scientists claim that they have found bacteria that have been in the endospore stage for 250 million years. So you kind of think about that as like a bacteria hibernating for millions of years. It is certainly possible. The 250 million mark is definitely under contention, but the fact that they can stay, you know, in this phase for certainly quite a long period of time is definitely very interesting. Now, with a lot of bacteria, this can actually cause some problems though too, because let's say you're in a hospital, you can't simply boil instruments if you have bacteria forming endospores. You actually have to increase the temperature above the boiling point to be able to destroy them. And so that's something that's sort of like a practical matter that you know, you've probably learned. You know, if you want to kill the bacteria inside of a pot of boiling water, you have to leave it for a certain number of minutes, but even that necessarily isn't going to guarantee that they're going to die with some bacteria, although it certainly will kill most, and it will make your water safe. You wouldn't want to do that if you were putting somebody's life on the line with, you know, doing a surgery or something like that. So it can make things complicated in healthcare, and sort of it's an interesting thing that bacteria can do. Alright, so this is just a diagram showing you binary fission. You've already seen one of these before back when we looked at mitosis and meiosis, so this is just for you to refresh your memory. I invite you to pause it if you want to spend a little bit more time looking at it. The reason why prokaryotes are so important is because and the reason why we study them so in depth is because all of the different ways that other organisms make a living can be found in prokaryotes. So you may have heard of these terms in middle school, or when we were in grade 10, I believe we also talked about them as well. And these terms are all designed to sort of describe how things make a living. So organisms that are photoautotrophs, what that means is that they get their energy source from light, and they are able to make carbon using carbon dioxide in the air. So photo, light, autotroph, that means that they're getting their carbon, they're making their own carbon molecules from the air, as opposed to eating their carbon molecules like you or I do. Chemoautotrophs are organisms that get their energy source from inorganic molecules, and a good example of this are, you know, I think I gave you in your notes package a little handout that talks about bacteria that can, for example, use iron or sulfur as an energy source. So chemoautotroph is a prokaryote that would use chemicals for energy and get their carbon source from the environment, so CO2 again. These types of organisms often live in sort of interesting environments, for example, around deep sea vents. Okay, there's no light that reaches down that far, but organisms have to get energy from somewhere, and you know, there's not a lot of organic matter for them to eat, so what they do is they take these molecules that are dissolved in the water and they use those as their energy source. So that's a chemoautotroph, energy from inorganic molecules, and their carbon source, they are getting it from CO2 in the environment. Alright, a photoheterotroph is an organism that gets its energy from light, but that gets its organic compounds or it gets its CO2 from eating organic compounds. And there's not a lot of examples of this, but there are some prokaryotes that do that. Chemoheterotrophs are organisms that get their energy from eating organic molecules or organic compounds, and they also get their source of CO2 from eating those organic compounds. So we're a good example of chemoheterotrophs. We get our energy from food, and we get our carbon from food. So if we're thinking about all these different types of ways of making a living, they all originated in prokaryotes. You'll remember that, for example, we know that plants are also photoautotrophs. They're multicellular eukaryotes, but they're only photoautotrophs because at some point in their history, they went through the endosymbiotic process with the chloroplast. So remember, those chloroplasts were originally prokaryotic life, and that is what allows them to do what they do. And that originated in the prokaryotes. Remember that the first photosynthetic bacteria were cyanobacteria. So all of these forms of making a living, all the ways of getting nutrition and metabolizing molecules, this all comes from prokaryotes. So sorry, up above you see that there's also saprobes, which are organisms that decompose things, and you also have parasites, which absorb nutrients from living hosts. Those are both subsets of chemoheterotrophs. So they both are examples of that. Bacteria can also do something really cool. You know, one of the things that they are able to do is take nitrogen from the air in its N2 form and convert it to ammonia. You're going to see when we look at plants that this is super important because our bodies need amino acids to be able to build proteins. And one of the main things that's on an amino acid is the amine group, which is made up of nitrogen and hydrogen. We wouldn't be able to form amino acids if it wasn't for our consumption of other amino acids, obviously. But plants, what they do is they're able to take nitrogen from the air and convert it into ammonia, and then that ammonia is able to be used to make amino acids in plants. So the ultimate origin of all of the amino acids in your body is the nitrogen in the atmosphere. But you wouldn't be able to get that nitrogen from the atmosphere. Like, for example, you can't breathe in nitrogen in the atmosphere and make your own amino acids. You have to eat your amino acids from something else that's able to do that. And it just so happens that there's a symbiotic relationship that exists between bacteria that are nitrogen fixing and the plant roots, so that they're able to take nitrogen from the air, convert it into ammonium, and use it for building amino acids. And remember that there are bacteria that have different types of relationships with oxygen. Remember that in early Earth's history there was not a lot of oxygen in the air, in the atmosphere, and well, there was none in the atmosphere. And so the introduction of oxygen into the atmosphere was deadly for most prokaryotes. Remember that oxygen is very, very reactive. And so they had no defenses to the oxygen that started to accumulate in the atmosphere, and they died. The vast majority of them went extinct. However, some of their descendants still live. We talked about tetanus in one of the previous presentations, that it hides under the rust, and that's how it's able to survive. So that's sort of, it goes back to that point in time. Now, the organisms that cannot exist at all in oxygen, we call those obligate anaerobes. They are obligated to live in an anaerobic environment without oxygen. There are what we call facultative anaerobes, which are prokaryotes that can live in the presence of oxygen or in the absence of oxygen. They can go both ways. It doesn't really matter for them whether there's oxygen around or not. An example of a facultative anaerobe that is not even a prokaryote, but that's very important for us, is yeast. It can survive in the presence of oxygen, and it can also survive in the absence of oxygen. So it would be a facultative anaerobe. Although it's not a prokaryote, I hope it gives you an idea of what those organisms would be like. And then you also have obligate aerobes, which are prokaryotes that need to survive or need the presence of oxygen in order to survive. So we've got a bunch of different ways of making a living here, all important because we see them later on in the development of eukaryotes, and so that's why it's important, and that's why we look at this in depth.